This picture over here really illustrate the first point that I made from on earlier slides, which are the importance of epithelial barrier health and its role in reducing African swine fever virus infections. As you can see on the left side, when we have very healthy epithelial layers, we can potentially prevent the African swine fever virus penetrate through the epithelial layers and get into contact with its target cell, which are macrophage cells and the dendritic cells, showing here in blue and the purple. And potentially that will help us to reduce infections, or at minimum to increase the minimum infectious dose are needed by this virus to infect the pig. On the other hand, if we have some damage on this epithelial layer, whether that's a gastric intestinal damage, diarrhea-related damage, or a damage related to another virus challenge caused a lung lesion, for example PCV2 or APP, nevertheless, this will really increase the African swine fever virus penetration into the donor layer of the epithelial layer and have them to get in contact with its target cells. And this really increases the chance of African swine fever virus infections, even by a low dose exposure. So the importance here is we really need to focus on the epithelial barrier health to reduce the African swine fever virus infections. There are some report or some paper published in the literature really support this idea. This is a paper published by Dr. Sandra Bloom's lab, who is a leading scientist in Germany on African swine fever virus. This is a paper published by her lab where they looked at the minimum infectious dose of African swine fever virus to unhealthy animals and immune compromised runt animals, where she concluded when she oral nasally administered 100 units of this virus, she could not infect a healthy pig. However, if she used even lower dose, 10 units, she was able to infect an immune-compromised runt pigs. And also, she mentioned in this paper that the first pig getting infected are having some gastric intestinal issues, which she think might contribute to this low dose infection of pigs. So from what we learned, or also from what we had from this paper, we can see the herd health and this mucosa integrity, especially the epithelial layer, its health, its integrity, and also the proper immune response are very keys to control this African swine fever virus.